Hello fellow students and welcome to this excellent video about SCPNs. No, not this heaven on earth but single-chain polymer nanoparticles. For the rest of the video called SCPNs. SCPNs have a polymer backbone with functional groups. These form intramolecular interactions that fold the molecule. Just like the Lego blocks of life. Proteins. When designed and synthesized properly, SNPNs can mimic the function of proteins. This is very interesting because proteins are very hard to synthesize in a lab. The main method of in vitro protein synthesis is called cell-free synthesis. This method is very time consuming, and because the DNA is in the cytoplasm it gets degraded over time. This causes defects, and with defects proteins have a good chance of losing their functionality. Just like indicators on a BMW. Letting bacteria do the hard work is also not perfect. Because bacteria have other needs than humans, they make different proteins. For one, bacteria rarely perform glycosylation of their proteins. This is an important step in making the protein function in the human body. Another problem with synthesis of proteins in bacteria is that there are often multiple steps of preparation and purification needed to make the protein function. These steps are very expensive, and that is why 200 microliter of DNA polymerase cost more than this amazing yellow sports car. To synthesize SCPNs, you first need to have a monomer that can be easily polymerized and functionalized. Methyl methacrylate, also known as MMA, has these characteristics. Therefore it is often used in the synthesis of SCPNs. The ease of polymerization is due to its easily split pi bond between the two carbon atoms by a free radical. A process often used is called raft polymerization. RAFT stands for reversible addition fragmentation chain transfer. With this method, the monomer is reacted with a raft agent and a radical source. The raft agent acts as an intermediate in the reaction and controls the polymerization of MMA by regulating the amount of free radicals in the mixture. The functionalization is done by changing the methyl group on the oxygen for something else more functional, like a benzene ring or a hydroxyl group. Changing to an hydroxyl group can be done by using palladium on carbon as a catalyst and hydrogen. The functionalization is important because this controls which functional groups are present in the molecule, and therefore the intramolecular interactions. To mimic proteins, you need to copy their 4 levels of structures. The primary structure is their amino acid chain that is being held together with peptide bonds. The secondary structure are mainly made out of alpha helix and beta sheets, which are being strapped together by hydrogen bonds. The tertiary structure is formed by intramolecular cross-linking such as ionic bonds, disulfide bridges, hydrophobic interactions and hydrogen bonds. The cross-linking folds the protein in a specific way so that it gains its function. The quaternary structure is formed by multiple individual proteins bonding together to be functional. For example, everybody's favorite protein, hemoglobin. It exists out of 4 subunits that are glued together by electrostatic forces. To mimic these 4 structures, you need to have great control over the polymerization and functionalization process. To achieve this, a raft-Passerini multi-component reaction is used. This is a variation of the raft polymerization to have greater control over the functionalization of the chain. The raft is the same as previously explained, but the Passerini reaction adds selective functional groups to the chain within reaction involving 3 different compounds. The added functional groups have the ability to form intramolecular interactions. These interactions fold the particle and this controls the shape and size. Those characteristics are important for the biocompatibility and function of the SCPNs. The thickness of the SCPNs plays a great role in the distribution of the particles in the body. SCPNs smaller than 6 nanometer are generally filtered out by the kidneys. This makes these particles very biocompatible but limits their time in the body. If you want the particles to penetrate the blood-brain barrier, they can't be much larger than 15 nanometer. Nanoparticles smaller than 200 nanometer like to camp in the liver, but particles smaller than 70 nanometer are mainly found in the spleen. The long-term effects of these particles accumulating in these organs is not yet known, but it is most likely. Everything has problems except this fantastic car. The main problem of SCPNs is that it is not possible to functionalize each monomer in the chain. Proteins are synthesized this way, and this makes it possible for them to form incredibly complex structures. Because of the lack of control, it's not possible to mimic conventional proteins with SCPNs. To make this happen, better methods of the polymerization and functionalization process have to be developed. Other problems with the current methods to synthesize SCPNs are that they are very complex, time-consuming, and prone to defects. For commercial use, it is more efficient to use the cell-free method and those pesky bacteria for the production of proteins. This doesn't mean that SCPNs are not functional right now. They can be used to encapsulate drugs to facilitate controlled drug delivery, and also as a contrast agent in magnetic resonance imaging. Because of its potentially small size, it can be renally cleared and thus can't do any long-term harm. For this to work, gadolinium centers have to present in the particle. This can be achieved by changing the methyl group of MM8 or azide group to enable the azide cross-linking. In conclusion, it can be said that SCPNs have a bright future with multiple applications like protein mimicry, controlled drug delivery, contrast agent, and many more. For all this to happen, great advances have to be made in polymer chemistry. Because right now it is a great idea but not commercially viable, just like this year's YouTube Rewind. I hope you enjoyed the video and learned something useful. If you have any questions, you can ask them right now. And finally, here are the sources for the information in this video to prove that not all of this is complete nonsense.